I'm Becky Bradford, Senior Vice President for ATCC Federal Solutions. I'm so proud of the work this team has done, which resulted in being recognized for the NIH's Replication Challenge Award. At ATCC for over 100 years, our mission is to enable reproducible science by providing authenticated, high-quality biological resources and standards. For this award, the team solved a critical gap in reproducibility through standardized key starting materials for infectious disease research. This led to trusted outputs. Thank you to the NIH for continuing to promote the importance of reproducibility. Our approach at ATCC will continue to be focused on the advancement and the credibility of science.
One hidden driver of poor reproducibility in preclinical virology is the viral starting material. Replication is not only about repeating work. It begins upstream with the challenge material. Viruses change rapidly, and repeated passaging or varied growth conditions can introduce genetic changes that alter infectivity, virulence, and drug response. When that happens, comparing data becomes difficult and confidence drops as programs move toward the clinic. Our team worked to close that gap by putting replication at the very beginning.
We define replication as the ability to reliably produce viral challenge material with the same phenotypic, genotypic, and functional characteristics each time. To achieve this, we use a systematic approach. We first optimize the virus propagation conditions to reduce genomic drift, which we monitor using our informatics pipeline that looks beyond the consensus sequence and then characterizes any minor variant populations that may be present. We then confirm reproducibility of these conditions through engineering runs and then scale up production through controlled SOPs.
This approach led to measurable improvements in reproducibility and scale. Across more than 30 production runs for high-consequence viruses such as Lassa and Nipah viruses, we achieved consistent genomic integrity, stable titers, and lot-to-lot interchangeability. A major barrier for us was variability introduced both during propagation and downstream user passaging. This was overcome by standardizing production parameters and introducing an out-of-the-vial model, which removes uncontrolled steps and dramatically reduces experimental variability.
Our replication strategy is sustainable because it's built into ATCC's standard operating procedures, so it can outlast any single project, and it scales across virus families. By depositing well-characterized challenge materials into NIAID's BEI Resources Program, we enable broad access for academia, government, and industry teams worldwide for these ready-to-use critical materials. When we normalize replication at the input level, we can avoid variability, strengthen the rigor, thereby accelerating countermeasure development, all of which improve our preparedness for outbreaks. We see this model as a transferable blueprint for reproducible preclinical research. We at ATCC understand that in science, trust is built on what others can repeat.
Clinical research today has a reproducibility gap. Even when studies are well designed, the actual analytic workflows are highly customized and often partially documented. That means translating a published method from natural language into executable code is slow and labor intensive. So in practice, replication becomes the bottleneck. It's not that we don't value replication, it's that the process itself does not scale. To address this, we developed LATCH, which stands for LLM Assisted Testing of Clinical Hypotheses. The key idea is a hybrid system. We use an LLM only for what it's good at, interpreting ambiguous natural language methods and mapping them into structured plans. Then, all statistical analyses are executed by a deterministic engine in R and Python, so analyses are reproducible. Ultimately, the framework translates clinical research methods from text into executable code, covering study interpretation, variable selection, cohort construction, and statistical analysis, while capturing every step in an analytic report to enable transparent and auditable replication. Importantly, this approach is privacy preserving because the model does not access patient level data. It operates only on the database schema, such as table and variable names, without exposure to sensitive information. We evaluated this approach by replicating 20 peer-reviewed studies using the enhanced dataset. We observed overall agreement across key metrics, including cohort size, effect direction, and statistical significance with some expected variability. Importantly, we reduced replication time to just 2 to 20 minutes per analysis. Latch addresses 3 key barriers in replication. First, it addresses underspecified methods by logging every step from code to results. Then, it also replaces slow and manual replication with a scalable LLM-assisted workflow with human review. And third, it reframes discrepancies not as failures but as auditable, actionable insights. Together, this makes replication faster, more transparent, and more informative. The bigger picture is this. If we lower technical barriers through automation, replication can shift from a high-cost exception to a routine process. Because Latch is model agnostic and schema-based, and it can transfer across datasets and workflows with the proper setup, which involves standardization of data and schemas. So ultimately, this is about normalizing replication, making it a standard, scalable part of how we do biomedical science. Replication does not have to be exceptional. It can be built into the infrastructure. Thank you.
Hello, my name is Michele Di Mascio, and the replication work presented in this submission was conducted while I was at the National Institute of Allergy and Infectious Diseases. Our work addressed a key question: can antibody-based probes reliably image lentiviral replication in vivo? This matters for understanding HIV reservoirs under therapy, but also reflects a bigger challenge in biomedical science, as no wild-type virus had been previously successfully imaged in vivo. Over the past decade, influential studies suggest that this was possible, but key validation steps were missing. So our goal was to rigorously test whether these findings could be independently reproduced. We reproduced the original experimental conditions as closely as possible, including the same antibodies, radiolabeling methods, infection models, and imaging parameters. In parallel, we introduced the critical validation steps that were absent from the original studies, including ex vivo competitive binding assays to confirm specificity, autoradiography, and probe stability analysis. We also tested alternative probes to further confirm the robustness of our findings. Across all approaches, our results were consistent and unambiguous. We found no detectable GP120 specific signal either in vivo or ex vivo, including in lymphoid tissues such as lymph nodes and spleen that are known to harbor the highest levels of viral replication. These findings define clear limits of antibody-based viral imaging and offer a more realistic framework for future studies. They also highlight the need for critical evaluation in imaging and HIV cure research communities, as new methodologies can be quickly seen as established. Key barriers included limited validation and challenges in publishing negative results. We address this through rigor, transparency, persistence, and independent replication. This work highlights a key principle: validation must come before translation. Early ex vivo checks can prevent costly downstream research. In complex, resource-intensive research, independent replication is often limited, highlighting the need for greater transparency and coordination. I would like to thank the NIH for supporting this important initiative focused on replication.
On behalf of our team from Deloitte Consulting LLP, I am honored to accept this award. We are grateful to the NIH Common Fund for recognizing work that elevates replication and reproducibility, as those principles are essential for strong, credible science. Our submission focused on a simple but important idea. In biological research, results need to be consistent across laboratories if they're going to inform public health decisions with confidence. For our submission, we focused on the efforts by the influenza community to ensure fidelity of their findings. Influenza remains a major public health threat because of its seasonal impact and its pandemic potential. That makes it critical that the assays and models used to assess risk and immune response are not only scientifically rigorous, but also reproducible. The studies highlighted in our submission showed that while some variability in virological research is unavoidable, much of it can be reduced through standardized protocols, shared reagents, normalization to standards, and cross-laboratory evaluation. They also demonstrated the value of testing reproducibility in real-world settings across both domestic and international networks of researchers, institutions, and laboratories. What makes this especially meaningful is that the impact goes beyond methodology. These assays and animal models support influenza risk assessment and pandemic preparedness. Improving their reliability helps strengthen trust in the evidence that researchers, funders, and public health leaders use to make consequential decisions. We also want to recognize that this work reflects the power of collaboration. The progress described in this submission was made possible through sustained collaborations and research networks, especially FluCop and SEER, and through the commitment of scientists who are willing to work together to improve how research is done and not just what research finds. We'd like to thank Richard Webby's laboratory for providing the publications used in our submission, as well as all of the investigators, authors, and participating laboratories who contributed to the studies. We hope this recognition helps reinforce an important message. Investing in replication is investing in better science, better preparedness, and better public health outcomes.
Hi, my name is Catherine Groff, and I am a scientist at PETA. The replication exemplar that I'll be presenting, Advancing Replicability in Biomedical Research Through the Integration of Recombinant Antibodies, brings together the multidisciplinary team listed here. The core problem we set out to address is unreliable, poorly defined antibodies. As widely used research tools, antibodies have historically contributed to irreproducible results due to batch variability and undefined composition, as well as poor validation. Our team sought to address this gap by integrating sequence-defined recombinant antibodies as a systemic solution to improve replicability at its source. This slide summarizes the key initiatives within our replication exemplar, which span 3 key dimensions. First, we supported the development of new recombinant antibodies for widely used and clinically relevant targets. Second, we prioritized rigorous characterization of these antibodies with transparent dissemination of methods and results. Third, we integrated recombinant antibodies into university laboratory curricula, training the next generation of scientists to routinely use reproducible reagents. Outcomes related to replicability in the context of transitioning to recombinant antibodies are evaluated based on the intrinsic properties of the new reagents and standard antibody characterization and reporting information, as described on this slide. Capacity was a major barrier we encountered. Millions of antibodies are available for purchase from hundreds of suppliers around the world, and securing funding to evaluate reagents and develop recombinant antibodies is necessary. To address this barrier, these initiatives have been undertaken in a collaborative nature, and team members had to prioritize which antibodies to develop and characterize and which to use within studies and practical courses. Building on this momentum, this team plans continued collaboration and targeted initiatives to develop, characterize, and apply recombinant antibodies. This team will continue to expand the availability of recombinant antibodies within commercial and academic catalogs. And to further enhance transparency and confidence in recombinant antibody use, stakeholders will publish characterization data, including through dedicated venues such as antibody reports, and support open access dissemination via resources like the University of Geneva's Sequenced Antibody Database. Finally, we aim to replicate and disseminate scalable educational models. For example, the Technical University of Berlin's course materials on the use of recombinant antibodies is openly available for adoption by other universities. The key takeaway is that replication can be normalized by integrating reliability into foundational research tools. The diversity of stakeholders participating in initiatives to integrate recombinant antibodies highlights both the critical role of reliable reagents in advancing research replicability and the widespread opportunity for for researchers across sectors and disciplines to integrate the use of recombinant antibodies.
Hi, my name is Elias Washington. I work in translational and clinical research with my team, Leonid Saad. Together, Team C20D3 Vitamin A Replicate. Our work focuses on retinal degeneration. Our central question was, does vitamin A dimerization play a causal role in retinal degeneration? And if so, could slowing vitamin A dimerization preserve retinal function or prevent retinal degeneration? During the visual cycle, vitamin A is continuously used in photoreceptor cells. A small fraction of vitamin A reacts with itself to form dimers that accumulate in the eye. The biological consequence of this reaction was not fully understood. So the question became, does vitamin A dimerization contribute to retinal degeneration, or is it simply a byproduct of normal physiology? To answer this question, we developed a chemically modified form of vitamin A, C20D3 vitamin A. C20D3 vitamin A selectively slows vitamin A dimerization through a kinetic isotope effect, while preserving the normal biological function of vitamin A. C20D3 vitamin A allowed us to isolate a single variable, the rate of vitamin A dimerization, and directly test the extent to which vitamin A dimerization is causally linked to retinal disease. We tried to answer this question using multiple biological systems, including mouse, rat, swine, and human populations in various stages of retinal degeneration. No single model and no single patient can capture the full heterogeneity of human disease, which is why replication across systems and patient populations was essential. Our definition of replication was independent, multimodal verification of the biological consequences of impeding vitamin A dimerization, using complementary endpoints, morphological, biochemical, and functional. This replication work was conducted across more than 20 independent sites globally. Across all systems, we observed a consistent relationship. The faster the rate of vitamin A dimerization, the worse the retina outcomes. Together, these findings support a causal role for vitamin A dimerization in retinal degeneration, and a possible means to intervene therapeutically. This level of replication strengthened, increased confidence in the findings, and enabled translation from mechanism to possible therapy. Replication isn't something you claim once, it's earned and never ending. Thank you.
On behalf of the Conduct Science and Maze Engineers team, we are honored to accept Track 2 of the NIH Replication Prize in Preclinical category. I'm Louise Koskaden, Director of Science and Development at Conduct Science, presenting today on behalf of our team. Conduct Science is a science manufacturing company. We were founded in 2013 to build the equipment, software, and publishing tools that help research labs run the same experiment the same way and get the same results. Our products are used in more than 1,200 research institutions across 50 countries and cited in over 280 published studies. The prize recognizes 13 years of that work. We make a wide range of research equipment and software, behavior apparatus, microfluidic chips, histology, and microscopy systems, computer vision software, lab procedure automation, and a publishing platform for scientific methods. Every product we make is built to match the original published study that defined that method, and every change to a product is recorded with that date. For this prize, we documented what it looks like in our most established area, which is behavioral neuroscience. Maze Engineers makes the physical equipment. Conduct Maze is the software that runs the equipment. Conduct Vision uses computer vision to watch the animal and produce the same numbers in every lab. Our technology transfer program turns inventions made in academic labs into products that other labs can buy. And conductscience.org publishes the full method online, with the build files that anyone can download and reuse. The proof is whether different laboratories get the same results when they run the same experiment. In 1999, a study by Crabbe and colleagues ran the same mouse experiment in different labs and reported a measure of how much the results varied across sites. Their value was 0.76. Across 58 independent labs using our equipment, the same measure is 0.41, about half of what they reported. That means much more consistency between labs. Our equipment has been used in more than 1,150 research organizations in over 50 countries and is cited in 280 published studies. The barrier we kept hitting was trust. Academic labs do not buy from a vendor without proof. We close that gap by making every citation, every build file, and the full change history public for every product we ship. That public record lives at conductscience.com/verified. Behavioral neuroscience is one area. The same approach applies to many other parts of science where labs struggle to repeat each other's work, including microfluidics, histology, and imaging. Match the equipment to the published paper, run the experiment in the same way every time, track every change. Share the files anyone needs to do it themselves. Replication is a problem we already have the tools to solve. Scaling that across biomedical research is what the next decade of Conduct Science is for. Thank you.
Hello, I'm Magdalena Cassandra. I'm a Director of Research and Development at the Center for Stem Cell Organoid Medicine at Cincinnati Children's Hospital. My team focuses on de-risking human organoid platforms for drug discovery and development. While organoid models have a tremendous potential to improve how we predict human drug responses, they remain difficult to reproduce, hard to scale, and are often not deployable beyond expert labs. So the question we asked was, can we turn this promising biology into something reproducible, scalable, and fit for real-world applications? Our goal was to address this replication gap not only by reproducing a high-impact liver organoid model for drug-induced liver injury detection, but by transforming it into a standardized, scalable, and translational platform. We approach replication as a design problem instead of copying the original workflow step by step, and we focus on preserving what truly matters, its biology, while re-engineering everything else to improve control, consistency, and scale. First, we reconstructed the published assay to establish a biological baseline. Then we redesigned the system. We moved it to matrix-free conditions and introduced microcavity-guided aggregation to control organoid formation. Next, we automated the workflow end-to-end to eliminate user-dependent variability and enable reproducible execution. We validated its performance across multiple donor lines and differentiation batches. And finally, we integrated AI-based phenotyping to enable high-content, non-terminal toxicity assessment. This resulted in real quantitative improvements. We reduced interline variability by approximately 90% and intraline variability by about 40%, demonstrating a substantial gain in reproducibility. We scaled production to support up to hundreds of thousands of organoids per run, with workflows validated across more than 30 donor lines and multiple differentiation batches. Importantly, we preserve biological fidelity, maintaining compound-specific toxicity responses and mechanistic injury signatures consistent with known DILI phenotypes. The major barrier in organoid field is high variability and user dependence. We address this through controlled aggregation, multi-stage quality control, end-to-end automation, and AI-based phenotyping, allowing us to finally separate true biological signal from technical noise while increasing usability and transferability of the system. In conclusion, we believe that replication should not be an end goal. It should be a foundation. We showed that it's possible to faithfully replicate biology while engineering in reproducibility and enabling deployment. Importantly, this approach is designed for adoption through standardized workflows, automation-ready systems, and shareable organoid precursors. By normalizing replication, we build the foundation required for cross-sector validation needed to move organoids and other new approach methodologies from promise to practice. Thank you very much.
Thank you so much for the recognition of this award. We are the Resi team. My name's Simon from Vanderbilt Health. We're a group of biostatisticians at Vanderbilt and Wake Forest who aim to improve replicability in neuroimaging with methods that focus on standardized effect sizes. Our work is motivated by challenges with replicability of neuroimaging research studies that have been recognized for over the last decade. We focus on standardized effect sizes because they're inseparable from replicability.
My name is Heidi. I'm assistant professor from Wake Forest University School of Medicine. Our research program addresses the replicability crisis in neuroimaging through three complementary strategies. First, we have developed a principal study design that improves the replicability of brain behavior associations, examining how study design features can be leveraged to improve the replicability of downstream neuroimaging analysis. Second, we introduced a generally applicable effect size index, RESI, to facilitate standardized effect size reporting in neuroimaging, helping researchers move beyond p-values toward more replicable inference. Third, we integrated three effect size report, this effect size reporting framework directly into neuroimaging workflow, enabling spatially resolved acceptance and rejection size that help distinguish scientifically meaningful effects from noise.
I'm Megan. I'm one of Simon's PhD candidates. We address multiple barriers in our work. The first is that effect sizes are incompatible with conventional neuroimaging analysis, which focuses heavily on hypothesis testing. We also face the challenge of heterogeneity of existing model-specific effect sizes. The RESI is universally defined, so it can be applied across contexts and complements neuroimaging analysis. We designed the RESI R package to be accessible, making it easy to add effect size estimates and confidence intervals to common model summaries, as you can see here. To date, the package has over 12,000 downloads, and the RESI methods papers have over 150 citations in papers spanning multiple biomedical domains.
I'm Xinyu Zhang, Simon's PhD candidate at Vanderbilt. To conclude our presentation, the RESI can be used across scientific fields to report universal effect sizes and can be easily implemented with our R package. Requests for new models or features can be submitted through our website. More importantly, statistics is the science of replicability. Rigorous statistical practice is a defense against the replicability crisis, and our research improves replicability through effect size reporting, better study design, and statistical modeling.
Hello, my name is Kayi Yang. Today I am here on behalf of the SOAR team, which stands for Spatial Omics Analysis Replication Team. Our team consists of Cecilia Young, Jocelyn Bright, Kimberly Smythe from the Fred Hutch Cancer Center, as well as my colleagues from University of Washington, Ling Hong Hung and Bryce Fukuda. Today, I'm going to present to you software tools that facilitates reproducible analysis of imaging data. And spatial proteomics provides a spatially resolved view of protein expression and localization within tissues and cells. Analyzing these complex data involves multiple steps and multiple software tools. So we present to you a graphical, containerized end-to-end workflow consisting of modular widgets. So the first step of this workflow includes starting QPath and reading in the image data. Then we are going to perform segmentation within QPath, and the results of segmentation will be exported as a text CSV file. And this segmentation results will be read by a Jupyter notebook to perform unsupervised clustering. And on top of that, we will also generate visualization results such as UMAP and heat maps. And these clustering results will be imported back into QPath. Clustering results will be overlaid on top of the original tissue image. And we have tested our tool on using human colorectal cancer tissue. So what we're looking at is a UMAP visualization of our clustering results in reduced dimensional space. And the dots represent the cells and the colors represent the clusters. And what we are looking at here is the clustering results overlay on top of the tissue image. And notice that the same color corresponds to the same clusters between the two images. Our clustering results has been evaluated by two pathologists, and we found that the cell types to be biologically relevant. And following cluster validation on the image, parameters can be iteratively modified using our graphical user interface, and the analysis can be rerun, enabling reproducible and iterative analysis. A quick recap. So we have shown you an end-to-end analytical pipeline that capture interactive graphical user input to facilitate reproducibility and interoperability of research involving imaging data. We allow integration of third-party tools such as QPath and Jupyter. Our tool is open source. At this link, you're going to find the source code, user documentation, a demo video, as well as a pre-configured GitHub Codespaces instance in which you can test drive our tool without installing any software. So what's next? We want to extend our tool to support additional image and multimodal data types, and we want to facilitate adoption of our tools and build a user community. Thank you for your attention.
Hello, I am Cassandra Johnson-Neely, and I've worked in the clinical research field for over 20 years now. During that time, I've worked in several different indications, such as metabolic disease, cardiovascular conditions, and infectious disease. One thing that's been clear throughout my career is that research just doesn't stop in the journals. It directly impacts how our patients are being treated on a day-to-day basis. And that can only work if the data behind those decisions are consistent, reliable, and holds up over time. At its core, replication is about making sure results are consistent and can be repeated, not just observed once. The gap I focused on is that replication is often treated as something separate instead of something we can build into daily research operations. My approach was simple and built into everyday workflow. We used dual verification for key outcomes, tracked protocol deviations more consistently, and compared results across similar studies running at the same time at our site. What makes this approach different is that replication wasn't treated just as a separate activity. It became part of how we already worked. After implementing this, we saw fewer data discrepancies and we were able to identify issues earlier instead of waiting for monitoring visits. One of the biggest challenges was time because site teams, are already managing a whole lot on a day-to-day basis. So instead of adding more work, we integrated replication into the processes that was already there, the existing processes, which made it both practical and sustainable. What it showed is that replication doesn't have to be complex or resource heavy. It can be built into everyday research operations, especially at a site level. And when it is, it helps improve data quality, consistency, and trust in research, which ultimately benefits patients. Thank you.
Hello everyone, my name is John Worsley from Mangada. Today I'm presenting our research on how high-altitude hypoxia modifies the efficacy of ACE inhibitors. Our team focused on a critical gap in cardiovascular pharmacology, the assumption of environmental neutrality. Most life-saving medications are validated in sea-level labs, yet millions of patients live or travel to high altitudes where oxygen levels drop significantly. We sought to address the lack of replication in this field, specifically looking at how the environmental noise of different microclimates lead to contradictory data regarding drug performance and patient safety. This is a translational, clinical, and preclinical research study. The replication strategy. To resolve these discrepancies, we implemented a three-tiered replication strategy. First, we moved beyond the single-site lab by conducting multi-site replication across an elevation gradient in the Donner Lake Basin. This allowed us to find the stability boundary where a drug's effects begin to change. Second, we integrated environmental metadata. By logging real-time oxygen, pressure, and UV data, we were able to mathematically subtract ambient noise from the actual biological signal. Finally, we used digital twin modeling. Instead of predicting all subjects as a monolithic group, we modeled each participant's specific oxygen history. This innovative approach allowed us to account for acclimatization memory, and that would bring about results that are far more precise and reproducible than previous studies. Impact in overcoming barriers. The impact of this high-altitude work is significant. Over 180 million people live in or visit high-altitude regions. For them, an altitude-adjusted dosing protocol is a preventative measure against stroke and cardiac events. The primary barrier we overcame was the issue of lab weather, the unrecorded environmental variables that often make biomedical trials fail. By standardizing high-resolution metadata, we increased the rigor of our findings and turned the environment from a nuisance variable into a predictable pharmacokinetic factor. This reduces healthcare waste and provides a more rugged framework for future clinical trials. Conclusion and future outlook. In conclusion, we must stop using the environment as a backdrop and start viewing it as a vital organ in the pharmacokinetic process. Our rugged medicine framework is designed to be transferable regardless of the topography anyone would call home. Thank you.
Hi, I'm Lisa Bastarash, and I have the honor and privilege of presenting the Phenotype-Genotype Reference Map on behalf of Team PGRM. This is a tool that we developed to improve biobank data science through replication. Now, a biobank is simply a large cohort that links genetic data to a lot of phenotypic information across a population. And these biobanks have been phenomenal for improving our understanding of human genetics. But there are some challenges to using biobanks, and one of those challenges is that it can be very difficult to understand the quality of the phenotypic information that is contained within these biobank data sets. So we created a tool to attempt to address that gap. And we did this by creating a tool that allows you to assess phenotype data quality via replication. To do so, we first curated known genotype-phenotype associations. There are many of these that have been put out over the last 2 decades in the literature, and they have been curated through something called the GWAS catalog. So we mined the GWAS catalog and found nearly 6,000 associations for about 150 different phenotypes that we represented as phe codes. And these are all associations that you can attempt to replicate in essentially any biobank data set. We then benchmarked this tool by measuring what percentage of associations that are known and in the literature are replicated across 4 different mature biobanks. That includes BioView, Michigan Genomic Initiative, UK Biobank, and Biobank Japan. And finally, we released our code with all the methods and all the replication attempts on GitHub to encourage the community to use this tool for their own data sets. We've been very pleased to see that many have taken up this tool for purposes that we envisioned in our initial paper, as well as novel applications. So there have been a number of groups that have used the PGRM to evaluate the overall data quality in new biobanks, such as All of Us and the Taiwan Precision Medicine Initiative. There's also been uses of the PGRM to fine-tune phenotyping algorithms. And finally, there have been some very interesting creative uses of the PGRM to assess things like the effect of ancestry on replication or the evaluation and mitigation of inclusion bias. I'd like to thank my phenomenal team, Team PGRM, for all the work that was put into creating and benchmarking this tool. In the future, we hope to update the PGRM to include more recent GWAS associations that have been put in the catalog since we released the tool. And more broadly, we're hoping that more people will hear about the PGRM so that it really normalizes the reporting of these types of replication metrics in publications, which we believe will enhance the rigor and replicability of the results and of our field in general. So thank you so much for your attention.
Good afternoon, everybody. I'm Jagdish Ramasamy from University of Illinois at Chicago. I'm the Director of Bioscience Research, Hematology Oncology Division, College of Medicine. So first I would like to thank NIH for selecting me to get this Replication Prize winner. Today I'm very glad to share some of our findings and recent discovery with all of you. Today title of my talk is mitochondrial retention in red blood cells. Our landmark discovery is mitochondria in circulating red blood cells and reticulocytes of sickle cell disease patient. And this novel finding we published in Experimental Hematology in the year of 2017. And this paper is cited more than 90 publications, underscoring broad impact in hematology and mitochondrial biology. Here is a replication strategy and global validation across independent institute. So its replication was integrated into multiple NIH projects with pre-registered protocols and procedures to ensure consistency, and we integrated to our previous funded projects as well as our current and planned NIH projects. So this finding also applied to other diseases such as Rett syndromes, SLE, or spirocytosis, so on. So there are also common pathological cascade and broader biological implications. Here I would like to summarize the identification of mitochondrial retention in red blood cells of patients with sickle cell disease, SLE, Rett syndrome, spirocytosis, and other mitochondrial dysfunction-associated immunological and neurovascular degenerating diseases require for replication approaches multi-institutional, rigorous open science replication framework embedded across related NIH research proposals. I would like to highlight some of the key references here for you. I would like to acknowledge all our collaborators here, and also I would like to thank NIH. And finally, thank you so much for everyone listening this talk.
Hello, my name is Alexander Tropsha. I'm a professor at UNC Chapel Hill and co-founder of a UNC startup, Predictive LLC. Our team also includes Professor Eugene Muratov, also at UNC and co-founder of Predictive, and Ricardo Tiegi, a research intern on our team. Our academic lab has had a long commitment to addressing the quality and reproducibility of biomedical research. We have developed best practices for data curation and model validation that have been widely adopted by the research community. We are very happy to accept Track 2, NIH Replication Prize. Our research strategy is illustrated on this slide using developmental neurotoxicity, or DNT, as a use case of in silico NAMs. We start from comprehensive data collection, including laboratory and real-world data, followed by careful data curation. We integrate all collected data into biomedical knowledge graph using special curation protocols. We mine knowledge graph for connected knowledge about DNT to elucidate adverse outcome pathways and use key events in these pathways and screening data collected in vitro to build AOP-informed DNT NAMs models with an ultimate objective of achieving regulatory acceptance of these models. We promote 3 key pillars in support of reproducible, high-quality research. First, all data must be carefully curated using standard, transparent protocols to enable any meaningful analysis or use of this data. Second, all models must be externally validated using reliable, data leakage-free protocols for data division and training, test, and validation sets. Ideally, prospective validation must be included as part of model development in collaboration with experimental researchers. Third, in biomedical research, we must review endpoints and assays for their relevance to human health to develop relevant models. We have identified data quality, both in terms of chemical structure and biological measurements, as a major barrier on the path to reliable models. We have developed and shared multiple technical protocols that support rigorous data curation and model development. There are 2 key lessons from our research I would like to highlight. First, as we have stated in our key publications, in dealing with historical data and datasets, it is important to follow the trust but verify strategy and apply data curation protocols to any dataset, even considered curated by its developers. Second, universal adoption of data curation protocols and validated modeling workflows enables better science and supports regulatory acceptance of in silico NAMs. It is critical to publish and openly share best practices and tools for data curation and model validation, as we have done in our research. Finally, only through normalizing replication, when reproducibility is embedded from data collection through final validation, can computational evidence earn the regulatory and public trust it deserves. Thank you.